be a rare treat if you stick a gene into bacteria and the bacteria actually make the protein for you, if your protein has rare codons. That is, unless you're using a strain that has plasmids encoding the extra tRNAs that recognize those codons. So these are strains like DE3RPL or DE3RIL or DE3RP or Rosetta 2. These are all different bacterial expression cells that we can use to get bacteria to make proteins that use slightly different spellings than they're used to. So in the messenger RNA instructions for making a protein, they kind of spell which protein letters to be added. So these amino acids in the form of these 3-letter words called codon. So you have codon, 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 codon. Then the ribosome is going to go along, and the tRNA that has the transfer RNA is going to have the amino acid that matches the codon. It has an anticodon that matches the codon. And so as the ribosome is traveling along and making this protein, the tRNA is going to bring in the corresponding amino acid. So its anticodon is going to match the codon. The ribosome's like, okay, all cool, and then helps join the new amino acid to the growing chain that's making its way out of the ribosome. So all of this is happening, but there are multiple codons that can spell the same amino acid. So there are multiple spellings in the RNA sense for the same protein letter. And this means that we have redundancy in our genetic code. And different tRNAs will specialize because of their anticodon matching a codon. They'll have different tRNAs that recognize different codons, but that all carry that same amino acid. So although bacteria can have all of the ability to read all of the codons, they don't use them all equally. And the ones that they use more, they're going to stock up more on those tRNAs, because remember, the ribosome is going to have to wait for the tRNA to bring the corresponding amino acid. And so if you don't have very many of them, then there's not going to be them around to use it. Now, this doesn't come into play when the bacteria are making their own proteins that much, because the bacteria stock up on what they need for how they encode their proteins. But if we stick instructions for a foreign protein into those bacteria, like a protein that is with the genes from a human protein. So we might use one codon for arginine more than another codon for arginine, more than another codon for arginine, because there's a lot of them. So we would stock up differently, but now so that we can make the protein more efficiently. But when we stick it into bacteria and the bacteria is using the different, doesn't use that one as much, now our codon is like a rare codon. And so if we want the bacteria to make a lot of our protein, it's going to have trouble, because you're going to get stalls, because the ribosome is going to have to be waiting for those tRNAs that there aren't that many of. So one way that we can increase expression of a protein sometimes is by adding in extra copies of those tRNAs that'll recognize those rare codons. And this will allow us to keep the bacteria from stalling when they're making our proteins. And so there are different expression cells that you can use to express a protein in bacteria, and there are ones that have extra plasmids, so they have a piece of extra information outside of their own genome. So they have their own genome with their own DNA, and then they have a separate genome that has a separate plasmid, so the circular piece of DNA, and this is going to have instructions for making the tRNAs that are recognizing rare codons. So if you have an RIPL strain, that's going to have codons for arginine, isoleucine, proline, and leucine. If you have RIL, arginine, isoleucine, leucine, RIG, arginine, isoleucine, glycine, and then Rosetta, it has even more, this is a brand name, but it has this P-rare plasmid, or this P-rare 2 plasmid. And so these have different tRNAs for recognizing different rare codons for these amino acids, so that you don't have problems. So we've talked before about codon optimization, and this is a cheaper strategy that you can use if you're doing this in bacterial cells. And they're both strategies to try to get at this issue that our genetic code is degenerate. And so we have each amino acids has at least one tRNA, but they might have more than just one, and then different organisms are going to prefer one more than the other. So for example, this is from Novogen's Competent Cells Guidebook, and so these are showing the rare codons in E. coli. So the amino acid, the codon that codes for it, the fraction in all genes, and then the fraction in this class 2, and this class 2 is like their highly expressed genes. And so you can see that they have a lot. Some of these they barely have any, and some of these they use a lot. And so the idea with codon optimization would be to try to change your sequence of your protein to use the ones that the E. coli used more, or if you're doing it in a different type of cell, like so I would use, before I used codon optimization when I was expressing in insect cells, a human protein in insect cells, and I would change the sequence of the human protein so that I was introducing synonymous mutations, so basically making changes that wouldn't change the protein spelling, but did change the mRNA spelling. But so you can order things like that. But if you are doing it in bacteria, another strategy is instead of changing your stuff, you change those cells. And so this is what we're doing with these strategies, in which we add in extra copies of the tRNAs that recognize the rare codons. And I should note that tRNAs can get kind of confusing because there are like ones that recognize multiple things, and then there are multiple codons because of like the wobble position, the third base. There are also some tRNAs like where there are multiple things of the, like multiple copies of tRNAs that recognize the same anticodon or the same codon, and there's various complication things like that, and so I'm not going to get into it too much, but the basic idea is that if you add more copies of the genes for the tRNAs that are going to recognize those rare codons, you'll have a better chance of the E. coli successfully making the protein. I should know, however, that there are problems potentially. It's not always like, if you can have solubility issues and stuff if you try to get the cells to make too much of proteins. There's also, I read an article I'll link to about some potential problems with some of the plasmids. But this basic strategy has been used a lot, and it's been used a lot, so much that there are lots of different versions of it that you can purchase, with different variations on which extra tRNAs are included. So first off, though, let's start with what's our baseline. And so often our baseline is going to be a BL21 cells. So BL21 is like this strain of E. coli that's often used for recombinant protein expression. And so there's like the original basic version, and then you'll see a bunch of things in like parentheses after the BL21. And sometimes what's in parentheses is going to be things like RIPL or RIL or RP or P rare. These are going to be the things that give you the encoding, the plasmid. They have plasmids encoding the tRNAs for those rare codons, and we'll get more into those in a minute. But first of all, there's things that make these cells really good for this purpose, for expressing recombinant proteins. One of these is that they have mutations in these genes for proteases. And so there's like this ompT and then this Dlon. And so this ompT, so they have a mutation that prevents this cells from making this like outer membrane protease 7. And so this is a protease that these cells make, and they use it to degrade extracellular proteins, so things that are outside of their cells. When you go and you lyse open the cells, so you break them open to purify out your protein, you don't want them to have this protease there to chew things up. So this mutation is going to prevent that. Dlon, so basically this is another one of those proteases, but this is a serine protease called Dlon protease. And so normally it's tasked with degrading foreign or defective proteins. And so if you have a foreign protein you want them to make, you don't want them to chew it up. And so these are going to prevent it from chewing up your protein. There's also mutations to prevent it from chewing up your plasmid. So there's mutations in various methyltransferases and restriction enzymes that the bacteria use to mark their own DNA as like self and then cut up foreign DNA. Those methylation marks can sometimes also interfere with restriction enzyme cutting. And so, but when they have the DCM out, then you don't have to worry about it for some of them. And yeah, so this is going to help them prevent them from recognizing your plasmid as foreign and cutting it up, which is what the job of that system normally is. These strains also have a couple of other mutations that really don't have that much impact that I know on in terms of expression in this case, and so they don't have this F plasmid, this fertility factor or sex factor, and they don't have this galactose. They can't use galactose. One of the most common probably variations of BL21 that you're going to see is going to be this DE3. And this DE3 is going to indicate that the cells are going to make T7 RNA polymerase when induced. So it has the gene for the T7 RNA polymerase that's under the control of this LAC promoter. This LAC promoter is normally bound by this LAC repressor. But when you add IPTG or when you deplete it of their glucose and add lactose, then you can get that repressor. It'll bind to the repressor falls off, then you get the T7 polymerase made. What's cool about this is that if your plasmid, you have a gene on a different plasmid, so this is integrated into the host genome. So this is like this lambda DE3. This is integrated into the E. coli genome from this like phage, this bacteria phage. And so, but when it's in there, so then you have your plasmid, which has what you want expressed, and you put that in the cells. And if you put what you want expressed with a T7 promoter in front of it, now the T7 promoter, you're only going to get expression when you express T7 RNA polymerase, because this promoter tells this T7 RNA polymerase to bind there, and it's not recognized by the E. coli's own RNA polymerase. And so then when you express the T7 polymerase can bind, and then you can get your protein made. And so you'll see like sometimes you can get kind of leaky expression, and this can be a problem, especially if your protein is toxic to the cells. And so some of these strains that you'll see have this thing called P-lyse. This is going to express a low level of lysozyme that's going to inhibit the low level of T7 that's getting made when you don't want it. So the leakiness, there's leakiness here, and so you can get some T7 made. And then if there's T7 made, then this can get, your protein can get made. And so this P-lyse is going to make this lysozyme that's going to inhibit the T7, so you don't get that. But it can also reduce the expression of your protein. So you don't want to use like a P-lyse strain unless you really you have a toxic protein or you're having problems with leakiness, but normally you don't really need that sort of thing. Also, sometimes they have like extra copies of the SLAC repressor gene. And a couple other common alterations, and then we'll get more in depth into the coding, the codon stuff. Rec A- cells, these are deficient in a homologous repair recombination system in the E. coli. So the E. coli uses, like, if they have a mistake and they're trying to fix their own DNA, they find parts that look the same and then like use them as like templates for the other and putting things together and things. And so it can end up shuffling things around, which you don't want. So some of these cells have this REC-A mutation. Some cells have an N-to-A mutation. This is going to make it deficient in another endonuclease, so another one of those nucleic acid chewers. This one is a non-specific one though. And E. coli normally keep it in their periplasmic space, so like in between their membranes. And this mutation can keep your plasmid from getting degraded by that. Okay, so those were some of the other modifications. And so now let's talk about these rare tRNA plasmids. So these are some of the common ones that you'll see. So I'll show you the product guide in a minute for some of these. You can see, so Rosetta, so Novogen cells is like Rosetta and Rosetta 2. These are going to, we'll get into those in a minute. And then there are also other strains that are just, you might, sometimes I think in Vitrogen or something has like Codon plus, but if they have this like RIPL, RAL, or RP, these are going to be telling you that they have rare tRNAs for things. And these are going to be provided on a plasmid or plasmids. And the importance of this is that you might have to add another antibiotic to select for the presence of the plasmid. And so we're using antibiotic selection here. So you'll have to add like 2 antibiotics. So you'll have the antibiotic to select for the presence of your plasmid of interest, but then you also need to worry about keeping the plasmid that was already in the cells and allowing that to grow. Although for some of them, like there's weird things where for this guy, you don't need to add the antibiotic when you're inducing. And to be totally honest, I actually, when I was expressing proteins before, I hadn't been adding this antibiotic when I was doing the protein expression, just when I was doing the cloning and all of that stuff, the second antibiotic to keep the plasma there. But I wasn't really worried about having rare tRNAs or that sort of thing. But in order to keep that plasma, you do need to add the antibiotic, except in this case. And that's like, you can see it in they have another way to make sure that it gets passed on without you having to worry about the antibiotic. But that's just for that second plasmid. And because this one has 2 plasmids because it has a lot of extra things. So this like RIPL, so the letters basically, they mean something. So they are telling you this has the rare tRNAs for arginine. So the R, isoleucine, I, proline, P, and leucine, L. And so this is good for organisms with GC or AT-rich genomes. And these are spread out on 2 plasmids. This one has the kanamycin resistance, this one has the chloramphenicol resistance. And so you're going to add chloramphenicol to keep this plasmid. And then this one has streptomycin or spectinomycin, but they say that you don't need to actually add this, at least in the induction part. And so, this is going to have 2 extra plasmids, and then you're going to have your plasmid that you put in there. But the plasmids are going to be present in these other plasmids are going to be present in the competent cells that you buy. So the competent cells are the cells that can take in the DNA, the plasmid that you're putting in there. So RIL cells, these have the arginine, isoleucine, and leucine, but not the proline. And so this is good for proteins from organisms with AT-rich genomes. So you can see it's recognizing things with a lot of like A's and U's. So U would be T would be the equivalent of the U in the DNA form. Then RP cells, these are good for GC-rich genomes. So these have rare tRNAs for arginine and proline. And again, these ones all have like kanamycin resistance. Oh, sorry, why do I keep saying kanamycin? Chloramphenicol resistance. And so you add chloramphenicol to keep them growing. And then the other one, the other common one are going to be these Rosetta cells. And so Rosetta cells, they have extra tRNAs for rare codons of arginine, isoleucine, proline, glycine, and leucine. So here we're adding glycine that we didn't have before. And these are encoded in these like P-rare plasmid in the case of the Rosetta original, and then P-rare 2 plasmid in the case of the Rosetta 2, which also has another tRNA for another arginine that recognizes CGG. And these are chloramphenicol resistant as well. I'm not exactly sure what's going on in these plasmids because it looks like they also have tRNAs for other, for additional. They also have genes for additional tRNAs, it looks like, in their plasmid, so I'm not exactly sure, and I tried to find out, but I haven't found out yet. And you can see that these two, these are like, they're showing that in the P lyse rare ones, they have that lyse, that lyse that we talked about, that makes the lysozyme that's going to inhibit the low levels of the T7. And then the P-LAC I rare, these are going to have this LAC I. So they're going to have a copy of the LAC repressor so that you're really tamping down on the expression. And so then they sell a bunch of different versions. So you can see, like, this is just Novogen's guy. This is like an older one, actually. But you can see that they sell a bunch of different strains and a bunch of different versions with like P-lyse, with the lac, black eye, and various things like that. And so, like, when you look in these manuals, you can see the different genotypes of these strains, different versions that you can buy. So this X, so these cells are good for, this is a version that is good for if you want to add methionine in, like, labeled methionine. So this X, basically, it has a mutation that makes it oxotropic for methionine. And so oxotropic means that you need to provide it. It can't make it itself. And so this is going to allow you to add in like a radio-labeled methionine or a selenomethionine and get them to use that instead. This can be good. Selenomethionine could be good if you're doing like crystallography and you want to do some like isomorphic replacement and that sort of thing to try to figure out your phase in your crystal diagram. Sorry, I'm not going to go into that here. And then radioactive if you want to track protein production or that sort of thing. Okay, so those were these types of strains. And then if you can buy like the Rosetta strains and various things like that. I do not work for any of these companies. And I will say that there's like, someone was telling me today that apparently some of them, that too much of a good thing might be a bad thing, and that you can actually have expression of, because you're not like selectively increasing the expression of your protein, you could be increasing the production of other proteins that then might inhibit your protein, and there could be potential problems with this P-lyse rare 2 plasmid. I haven't read this in depth, but it's just something I'll link to that someone pointed out to me. So I don't want to be like, just let you know that that's out there, as well as potentially problems if you're having it with problems with solubility, if you're having it make it too fast and that sort of thing. Because sometimes it's good to pause a little so your protein can full, but it makes more of an issue when you're trying to overexpress a protein and then the tRNA is missing. So in cells, actually, they kind of sometimes our cells use like rare codons because it allows kind of the ribosome to pause a little, allow things to fold, and give things time to do that sort of thing. So it can be regulatory, but when and the codon, the tRNA availability isn't typically an issue, even for the rare codons. But when you're trying to overexpress a protein and you need a lot of it, then that rare codon can become limiting. A couple last notes. So another common strain that's used is K12. This includes like Nova Blue strains, as well as Origami strains. These Origami strains, they have mutations in thioredoxin reductase and glutathione reductase. So this is good if your protein has important disulfide bonds. So glutathione reductase and thioredoxin reductase, they can reduce they play important roles in like reducing disulfide bonds. And so if you have disulfide bonds in a protein that are important for the protein to fold or act normally, serve as a dimer and that sort of thing, you can knock out these reductases in order to keep those crosslinks formed. These crosslinks are often present in proteins that are like secreted and that sort of thing, whereas our cells have a more reducing environment inside of them. Okay, so I think that's mostly it. Also be careful about the origins of replication, make sure that they're compatible with one another. Make sure you're using the antibiotics, all of those various things. So your plasmid should all have different antibiotics if you can select for something that has all of them. Okay, so I think that's basically it and hope that helped.